good afternoon. My name is Doris Valencia Pico. I am here to present to you the citric acid cycle and how we can calculate the end result of cellular respiration cycle, which produces how many ATPs. So here I made a simple diagram of the citric acid cycle. We have been learning in our class that glucose turns into pyruvate, which gives us two molecules of pyruvate, which when it enters the mitochondria becomes acetyl-CoA. We know that here two NADH and two ATPs are created. ATP, there you go. Then the pyruvate is transitioned into the mitochondria. We talked about how it enters this preparation step, and an enzyme CoA is added. Then the two NADH and the two ATPs are produced. We know that one carbon dioxide is going to be removed by carboxylation in the pyruvate dehydrogenase complex, in the E1, E2, E3 complex. Then the citric acid cycle occurs. We know that we have acetyl-CoA here. Then it turns into a four carbon fragment, which is called the oxaloacetate. The oxaloacetate combines with acetyl-CoA with the help of citrate synthetase. Here, there's the citric synthetase and forms citrate here. This is an irreversible step, as we can see. Citrate synthase is regulated by ATP production. So there's too many of the NADH. This citrate synthetase is going to be inhibited. If there's too much ATP, it's going to be inhibited as well. If also this succinyl-CoA enzyme that we will see around here will inhibit this enzyme to produce citrate. However, if we have ADP, too much of ADP is going to stimulate the citrate synthetase to produce more citrate. So we know that those enzymes regulate. This is a very regulated step right here, very watched for. The citrate then is converted into isocitrate. This is an isomerization reaction. So basically there's the same amount of molecules, but they change in location. This citrate converting into isocitrate occurs with the help of aconitase, which allows this to be a reverse reaction. You can see here. After the isocitrate converts, the citrate converts into the isocitrate, then this converts into the alpha ketoglutarate here, which is a 5 carbon molecule. So here we see that we lost the carbon, therefore it's lost in the form of carbon dioxide, or in the process of decarboxylation. Then this isocitrate is converted into alpha ketoglutarate by the help of isocitrate dehydrogenase. Here, an NAD plus turns into an NADH. This here, you'll see it's an irreversible step. So just keep in mind, here we have 2 NADH and 2 ATPs. Here 2 NADHs are produced in this step. However, there's a high amounts of ATP. When there's a lot of ATP around, the isocitrate dehydrogenase gets inhibited. But low ATP and calcium around the environment stimulates this enzyme. Then the alpha ketoglutarate dehydrogenase uses the NAD to turn it into NADH, which this process helps convert succinyl-CoA. Succinyl-CoA here is then converted into our friend here, succinate. In this process, one CoA is lost. This generates a little bit of energy to form the pi plus the GDT. When ADP steals the phosphate, which is what we've learned in our class, the substrate phosphorylation, another ATP is created. The enzyme in charge of this is called the succinyl-CoA synthetase, which stimulates the conversion of succinyl-CoA into succinate. Succinate. So it goes from here to here. So this is a reversible step. We have also an enzyme that is anchored in the membrane of the mitochondria called the succinate dehydrogenase. This enzyme turns the FAD into FADH2. In this step, which happens in the complex 2, converts the succinate into the fumarate. This is a one-way reaction. You can see that from now on, we still keep our 4 carbon. So we have another enzyme here that helps turn, we'll call it the enzyme fumarase. Uses a little bit of water into the reaction to turn the fumarate into the malate. Then the malate dehydrogenase turns NAD plus into NADH. So there's another NADH produced here. This is an irreversible step. But here we see how the malate turns back into the this molecule here, the the oxaloacetate. Sorry, I'm having trouble with my names. So in our class we have learned that all this process from the glucose to pyruvate, so one molecule of glucose turns into 2 pyruvates. Then this cycle turns gives us two FADH and two FADH and 6 NADH, as we learn in our cycle. But now, how do we calculate how many ATPs are resulted from all the cellular respiratory cycle or respiration cycle? So there's two ways that ATP is formed: through the substrate phosphorylation and also the oxidative or oxidation by electron transport chain. So we know that NADH and FADH2 combined together, they give us ATP synthetase. So there's a way how we can calculate here, and we're going to use our ratios here. So we know, let me cover this here. We know that for one NADH, 10 hydrogen ions are released. We know one ATP, basically researchers are trying to conclude, but they're still thinking that one ATP requires 4 hydrogen protons here, or ions. So here we do the ratio, and this one NADH will give us 2.5 ATPs. On the other hand, we have that 6 protons are needed for or containing one FADH, and the same ratio for ATP and for hydrogen ions, which one FADH would give us 1.5 ATP. So using these ratios, I created this table here. We can calculate So during glycolysis, we know that 2 ATP are yielded and 2 NADH. So we multiply our ratio of 2.5 times 2. We leave the 2 ATP alone, and 2.5 times 2 would be 5 of NADH. Now, we know that pyruvate oxidation gives us 2 NADH, 2.5 NADH, 2.5 from our ratio times this would give us 5. Now, from the citric acid cycle, we know that we get 2 ATPs, we get 6 NADH per our previous explanation, and we have 2 FADH. That's the total in our citric acid cellular respiration. So now we do our calculation again. We'll multiply. The 2 ATPs are left alone. The 6 NADH times 2.5 would give us 15, and the 1.5 times 2 would give us 3. So we add all this together, and it gives us 32 ATPs per 1 glucose molecule. This is how we can calculate our ATP production. However, we gotta keep in mind that different electrons produced by the NADH in glycolysis can be shuttled into the first protein complex or the third protein complex. So different number of protons will be pumped into the proton gradient. So the conversion factor might be different. So we calculate that maybe between 30 and 32 ATPs will be produced from the cellular respiration cycle. I hope my explanation was helpful, and thank you for your time.